Thank you all for joining our session on coexistence theory, and a huge thanks to Melissa for helping me organize it and for running our crazy hybrid session. I'm really excited for it. So, coexistence theory gives us a powerful set of tools to understand when and how multiple interacting species can coexist in a community. And it also gives us a set of tools to partition different contributions of fluctuation-dependent mechanisms in varying environments, and how those also contribute to species persistence and coexistence. That said, a lot of existing coexistence theory work tends to focus on annual species. This is true for both theoretical work, where annual species can be easier to model, as well as empirical work, where it's easier to do manipulations and get data out more quickly. Recent advances in computational methods, especially those spearheaded in the Elner et al. 2019 paper, give us different ways to use simulations to partition and look at coexistence, using either empirically derived or theory-informed parameters. These types of computational methods help to generalize coexistence theory work to more systems, including those that are frequently disturbed, a broader range of species, including seed-banking annuals, long-lived perennial trees or grasses, as well as a broader range of models, for example, species interactions like the Beverton-Holt model, which are more complicated to deal with analytically. In this study, we use this computational and simulation-based approach to examine patterns of coexistence between annual and perennial species under varying environmental conditions. We also use this approach to partition different variation-dependent mechanisms that contributed to persistence and coexistence of the annual and perennial species in our community. We looked at these patterns for both a seed-banking annual species, here with 2 life stages, as well as a perennial, similarly modeled with 2 life stages. Each of these were not interacting with each other, but with the same reference competitor of a non-seed banking annual. This let us compare their different patterns of coexistence and the mechanisms contributing to coexistence. For these models, we set up population growth models for each species with their density-independent rates of germination, survival, reproduction. And as you'll see, these look very similar in our perennial community, perennial with the non-seed banking annual community, as with the seed banking annual. We then added in different competitive effects, here represented by our different alpha terms, which can decrease the reproduction of the species, both intra and interspecific. And again, these are mirrored in our perennial species, thinking about how these effects take place. But the contrast with our perennial species is that the, quote, storage stage actually persists above ground, and so can itself have intra and interspecific competitive effects. And these can be both on those fecundity reproduction life stage transitions, as well as, for example, survival stage transitions. And so when we're thinking about perennial species, there's a lot more going on with our inter- and intraspecific species interactions that might affect our patterns of coexistence. We simulated our communities through patterns of 2 different types of year, here marked always as a tan year and a purple year. And there are different ways that we can think about how species might respond to these different types of years. So one example is a case where one year is favorable for one species and the other year is favorable for the other species in terms of their vital rates, here marked as lambda, but this could include fecundity, germination, survival terms. And in this case, we would expect that in the years that are favorable for these species in terms of their vital rates, that would also be the year that that species would exert stronger competition in each of their favorable years respectively. As a quick method summary, we then create a time series of these environmental conditions of these 2 different types of years. We designate one species our resident species and simulate its population through time. And then our other species here called the invader, which is not about origin, it's just the species that we're considering its ability to recover from low density, which is a necessary condition for coexistence. We take that species and at each time point we introduce it to the community, again at a very low density, and look at its growth rate through this time series. And then we take the average across that time to look at the low population growth rate overall. Getting into that partitioning, here we do this by allowing different types of those vital rates to either vary with the environment or to be held constant. So here on the top row, again, those are all of our density-independent vital rates, fecundity, survival, germination, etc. And on the bottom row is our competition terms, our alpha terms. So we split this down with different simulations, looking at what happens if those are held constant at their weighted average across time. That gives us a fluctuation-independent growth rate here in green, or if only those vital rates are allowed to vary with the environment, which gives us the partitioned component of the environmental variation on the vital rates through time. Here is delta lambda. We can also switch that, hold the vital rates constant, and look at the environmental effects and the effects of variable competition through time, delta alpha, and its effect on our full low density growth rate. And then the remainder primarily gives us the storage effect, which is the covariance of our vital rates and our competition throughout time. But it does also give us this residual term as well, which I don't want to spend a lot of time on because it doesn't become particularly important in either our methods or our analyses, but it is there. So results, what does this look like? So on this graph, our x-axis is looking at what the ratio of those two environments is over time. So at the middle point, 50-50, basically our time series is half tan years and half purple years, to all the way of the extremes with it being all one or all the other. And then we do these different simulations, allowing different parts of our species model to vary with the environment to do our partitioning and split them out. So our full low density growth rate is shown here in black, and then key partition is the storage effect, which is here always in that dark red color, the variation independent rate, which is green, and then our vital rates varying in light blue and our competition varying in orange. And so this is when we have a seed banking annual as the invader coming into a resident community, which is always our non-seed banking annual. And unsurprisingly, since we have opposite environments that are favorable, when the seed banking annual is in the environment that favors it, it does well. And when the other environment is more common, it doesn't do as well and would not be able to coexist. And I just want to highlight with this pattern that we see very, very similar patterns when our perennial species is the invader, again against a non-seed banking annual. And these patterns are primarily driven, our overall low density growth rate is primarily driven by the fluctuation independent growth rates. We can flip this around and make each of these stage-structured species, our seed banking annual and our perennial species, the resident, and bring the non-seed banking annual in as an invader species. And here we see, again, kind of as expected, the opposite. So this is the low density growth rate of the non-seed banking annual when each of these is the resident, and it does well in the environment that favors it, not surprisingly, through time. So this gets a lot more interesting when we think about the scenario where one type of year is favorable for both species in terms of their density-independent vital rates. So for example, a wet year versus a dry year. And maybe the magnitude of response to that change between year varies with our different types of species. How we think about how our competitive strengths would vary in this situation depends on what we're thinking of as our limiting resource. So in one case we might say, well, in a wet year, all of these species are growing bigger, and maybe they're competing for things like light or space or nutrients, and so that bigger growth leads to stronger competition. In the opposite case, we might say, well, in a dry year, there's less water available, and water is the limiting resource in this case, and so we would have more intense competition in that dry year. So opposite to the pattern that we're seeing with the lambdas. So getting into results here, going back to that first case where the year that favors the species vital rate also leads to stronger competition. And here's what we see with these different species being in the invader. So a reminder, in the absence of competition, this purple environment would be the most favorable environment for all of the species here being considered. But that's also where we have stronger competition. So what we see in terms of results, first is that we end up with very similar patterns with our seed banking annual species as with our perennial species. The growth rate when they're rare is largely affected by these environmentally mediated mechanisms, especially the variation in the vital rate, so that delta lambda term. And the optimal environment here would be different with the competitor present versus without it. Again, thinking about what about the cases where those stage-structured species are residents and it's our non-seed banking annuals that are invading. In this case, the storage effect when the perennial is a resident is particularly strong. In fact, we see that it's much stronger than it is for the seed banking annual as a resident. All right, and then when we flip it, so we have the purple environment would be favorable for all of our species, but we have stronger competition now with the unfavorable environment, so in that tan environment. And in this case, we get a really strong nonlinear environmental response in our full growth rate. So again, that's in black. And what I find really interesting about this is that the pattern we see is actually almost opposite to the variation independent component. So that would be if we just kind of averaged across all our environmental conditions and weren't considering environmental variation, that partition there in green. And contributing to this again, especially strong, is the fluctuation in the environment and the direct effects of fluctuation in the environment on the vital rates, so that delta lambda. But the storage effect is also a strong contributor as well. And once again, when we have these stage-structured species as the resident, that storage effect is much stronger, plays out much more strongly in the case for the perennial compared to the case for the seed banking annual. And I think there's a couple of reasons for this. One is that even when the perennial is kind of, quote, storing through its bad years, it still exerts competition on that invader, the non-seed banking annual invader. And so that is a negative storage effect that makes it harder to invade. And this has such a strength that in this case, if we look at our full low density growth rate, it's again nearly the opposite pattern of what we would see if we were just averaging across our environment. So environmental variation is really, really important here for understanding species persistence and species coexistence. So this just kind of wraps up my conclusion. Big takeaway, the storage effect is really important to consider in perennial species, even if it's more tricky to do so. And it's particularly important not for thinking about a perennial just invading into a community, but for the role of the storage effect when the perennial is a resident and other species are coming in or persisting or coexisting in that community. And finally, that these variation-dependent mechanisms can actually fully overwhelm variation-independent patterns and are really important to consider when thinking about our overall low-density growth rates. Thanks so much for my fantastic lab groups, my funding sources. Please reach out to me on Twitter or by email if you have any questions, and I am so excited for the rest of this session. Thank you.